Only recently has autism spectrum disorder been given the appropriate time, research, and funding to determine the developmental disruptions from which it occurs. The disorder is now rooted in science, which has allowed for many individuals to receive an appropriate diagnosis. As such, this presentation endeavours to reflect on the physiological components of ASD, to understand how the development of the neural network is affected. Then, the therapeutic use of transcranial direct current stimulation will be evaluated to determine the usefulness for treating ASD, whilst recognising limitations into this research does exist. ASD is recognised as a neurodevelopmental disorder, which affects an individual's empathetic behaviours. This may include both social and occupational dysfunction, as well as restricted and repetitive behaviours. Importantly, it cannot be linked to one aspect of pathophysiology. It is multifactorial, consisting of genetic, epigenetic, immunological, and environmental circumstances. It is referred to as a spectrum disorder due to the variety of severity and symptoms present among individuals. Diving into recently published research, Korshin and colleagues have stated that ASD stems from multiple abnormal prenatal developments, including disruption to the cell cycling, which produces excess cell proliferation and spontaneous disruption to neural maturity. Likewise, Hahn and colleagues have reflected on the theory that there is an excitation inhibition imbalance in the neural networks which support cognitive processes, resulting in the aforementioned characteristics. A recent review gathers evidence that shows an altered growth pattern on the brain of those with ASD, particularly in the amygdala and frontal cortex. This abnormal pattern of frontal cortex development has been linked to an increased number of neurons in the area, specifically in the dorsolateral prefrontal cortex. Now, transcranial direct current stimulation is a form of non invasive brain stimulation, which works by targeting neuroplasticity and cortical excitability through the sensoromotor cortex. The overall aim of TDCS is to shift the membrane of neurons toward either polarization or hyperpolarization. This is done by modulating the levels of GABA and glutamine in the targeted areas, crucial amino acids which act as inhibitory and excitatory neurotransmitters. As a whole, brain stimulation is a therapeutic technique which modulates nerve activity. TDCS is a specific type of brain stimulation, which is regarded as non invasive and painless. As TDCS increases neuroplasticity and affects a targeted area of the nervous system, it is considered to be neurorehabilitation. These changes in neuroplasticity can then be associated with changes to the brain's functional connectivity. Although it may seem evident, it is important to note that the outcome of TDCS has been shown to be different between neurotypical control groups and those with ASD. For instance, a neurotypical control group may reap benefits from the stimulation after just 10 minutes, where at least 20 minutes is required for those with ASD. As discussed, TDCS is focused on a target area, typically around the left dorsolateral prefrontal cortex. After the stimulation on this target area, it is believed that facial recognition and empathy based social interactions are accelerated, directly relating to ASD. To better understand these concepts, we can review research completed by Hahn and colleagues in 2021. The aim of their study was to evaluate how multi-sessional prefrontal TDCS paired with concurrent cognitive remediation affected the characteristics and symptoms of ASD. To do so, they had a sample consisting of 41 participants aged 14 to 21. Recapping their results, their study found that after a 1.5 mA TDCS was applied with the cathode focused on the left dorsolateral prefrontal cortex and the anode on the right supraorbital region for 20 minutes, participants reported a reduction in repetitive behaviors as well as improved social communication. With the reduction in repetitive behaviors and improved social communication, there was an enhancement in participants' ability to socially function, ultimately demonstrating the potential of TDCS to address certain social deficits. Research in psychology is a continuous task, with improvements constantly waiting to be made which commit to evidence based treatment. In 2016, Lafoucher and colleagues published a paper reviewing the guidelines for utilizing TDCS as a therapeutic tool. This review included the discussion of many diagnoses, from motor stroke to depression. When it came to completing a PubMed search with the keywords TDCS and autism, only 36 papers were identified, 6 of these being clinical studies totaling 64 patients. This is whilst the search for TDCS and depression generated 287 papers, with 988 participants. Thus, the question of why is posed. Why is there minimal research into a crucial developmental diagnosis? Now, in the study we discussed, completed by Hahn and colleagues, they recognized one of their limitations was that only those with ASD and an IQ over 60 were recruited. Unfortunately, through this process, an abundance of individuals with ASD are excluded. This includes those who are further along the spectrum, per se, with severe concerns of behaviour and the inability to communicate. To positively move forth, my direction is for there just to be more clinical trials, meta analyses, and discussions. Now more than ever, it is important to educate the public on ASD and bring with that suitable therapeutic techniques, such as TDCS, which link to improving aspects of executive functioning, ultimately improving an individual's quality of life.